Today we're going to be talking about mitosis, and mitosis is nuclear division. We're going to divide the nucleus in two. Now mitosis is part of a larger process called the cell cycle, which we talked about earlier. So before mitosis takes place, the cell has grown to its proper size, the DNA has been replicated, and we've made the necessary proteins required for nuclear division. So let's go ahead and get started with the first stage of mitosis. And we're going to break mitosis up into 4 stages. The first stage is going to be called prophase. The second stage is going to be called metaphase. The third, anaphase. And the fourth, telophase. So, what's happening in prophase? 3 major things are going to happen during prophase, and we'll number those 1, 2, and 3. Now, the first thing that's going to happen is the nuclear envelope, made up of the inner and outer nuclear membranes, is going to break down. And I'm going to demonstrate that with a dotted blue line. So that is the nuclear envelope beginning to disintegrate or break down. So let's go ahead and write that down. Nuclear envelope breaks down. The next thing that's going to happen is spindle fibers are going to form. These are protein complexes that ultimately are going to be responsible for separating the chromosomes into the 2 cells. So spindle apparatus forms. The last thing that's going to happen is the chromosomes are going to condense. They're going to get shorter, they're going to get thicker, they're going to get more dense. We want to make nice, neat, little tight packages that we can move around the cell without worrying about them getting damaged. And the easiest way to do that is to take the DNA and squash it down into a nice, tight, neat little package. So I'm going to draw 3 chromosomes. And they're going to be different sizes, and they're different sizes in the body also. There's one chromosome, here's another chromosome, and there's another chromosome. Each one of these chromosomes consists of 2 identical chromosomes connected together by a centromere. So that little dot represents a centromere. And the centromere is the point of attachment of these 2 individual chromosomes right here. These 2 individual chromosomes, we're going to call these chromatids. Each one of these is referred to as a chromatid or sister chromatid. 2 sister chromatids, 1, 2, are going to make up 1 chromosome. Now, until these sister chromatids officially separate, they're still referred to as chromosomes, 1 chromosome. Even though they're made up of 2, we don't consider them 2 until they officially separate from each other. So I'm going to draw 1, 2, 3 chromosomes here, but in a human cell we have 46. But for simplicity, we're going to go with 3. So the third one, chromosomes condense. Now all 3 of these steps are occurring at the same time. They don't occur in any particular order. They all occur at the same time. Once the nuclear envelope is broken down, we're ready to move on to the next phase called metaphase. In metaphase, the chromosomes are going to begin to line up in the center of the cell. And the center of the cell, we're going to give it a special name. We're going to call the center of the cell the metaphase plate. Also called the equatorial plane, because it's the midline of the cell. And it's an area. It's not a physical structure. It's an area. Just like the equator of the Earth is not a physical structure. It's an area. This is an area. It's the midline of the cell. Now, the way these chromosomes get to the midline of the cell is that this spindle apparatus grows long protein arms, long protein tubes called spindle fibers. And these spindle fibers literally push, gently push the chromosomes to the center of the cell. So these are going to be spindle fibers. And they're made out of protein. The next stage of the cell, or let's go ahead and write what happens here. Chromosomes line up on the metaphase plate. So chromosomes line up on metaphase plate. Once that's occurred, we're ready to begin the next stage, called anaphase. And anaphase is going to begin when the chromatids, the sister chromatids, separate. Once they separate, that marks the beginning of anaphase. So we're going to go ahead and draw our spindle apparatus here and here. And we're going to show the chromosomes moving to opposite poles or opposite ends of the cell. What's going to happen here is these spindle fibers are going to begin to shorten, get shorter. And as they get shorter, they're going to pull the chromosomes to opposite sides of the cell. Now you can see that these two are no longer attached by a centromere. And because of that, these are no longer going to be considered sister chromatids. They're their own official chromosomes at this point. So this would be a chromosome, and that would be a chromosome. So during anaphase, chromosomes move to opposite poles or opposite ends of the cell. Once they reach the opposite end of the cell, the pole, that marks the beginning of telophase, which is going to be our next stage. Now what I'm going to draw here is I'm going to draw a circle. And this circle represents the nuclear envelope. The nuclear envelope is reformed. The next thing, which you don't see, is that there's no spindle apparatus. The spindle apparatus has disappeared. It's broken down. So spindle apparatus breaks down. It's gone. We don't need it anymore, so we break it down into its component amino acids so that they can be reused to make other proteins that we do need at this point. And number 3, the chromosomes are going to relax. They're going to decondense. They're not going to be as tightly wound around the histones. The DNA is not as tightly wound around the histones as it used to be. So I'm going to try to show this as long, thin strands of DNA. So chromosomes decondense. Or we could say that they relax. The chromosomes relax. Now, at the same time that this is going on, a separate stage is occurring too. And I'm going to put a little asterisk down here because this is not officially part of mitosis, but it's occurring at the same time. And this is called cytokinesis. And again, as a reminder, let's write not part of mitosis. But it's occurring at the same time. Now we can also see this large indentation here and here. These indentations are referred to as a cleavage furrow. So we can tell that this is an animal cell, because with an animal cell, the cell membrane literally pinches, pinches, and pinches until the cell splits in two. And this is due to a protein contractile ring on the underside of the cell membrane. As that protein ring contracts and gets smaller and smaller, it drags the cell membrane inward until the cell pinches in two. Now this occurs in animal cells. Plant cells can't do that because they have a cell wall, which is rigid. So the same mechanism isn't going to work here for a plant cell. A plant cell is going to form a cell wall right down the middle of that cell. And that immature cell wall is going to be called the cell plate. And as we build this cell plate and build it, eventually we're going to separate that cell into two compartments, two individual cells. At that point, the cell literally just sort of breaks apart and we end up with two cells. So a plant cell and an animal cell, they actually divide. Cytokinesis occurs two different ways. In an animal cell with a contractile ring, which produces these cleavage furrows, and in a plant cell with a cell plate. And that's going to be it for mitosis.